a todos muito bem-vindos a esse quinto webinar do programa BIOTA-FAPESP. Desta vez, estaremos focando em questões mais voltadas à taxonomia e ecologia animal. Good morning and welcome all to this fifth webinar from the BIOTA-FAPESP program that is focusing on taxonomy and ecology and animal ecology. It's a pleasure for us to have you all here participating in this webinar. And I would like to remind all that if you have questions, they will be answered at the end of the presentations, and they should be sent to the email contato@biota.org.br. Questions will be replied only during the webinar, and the webinar will be recorded by the Agência FAPESP YouTube channel. It's a pleasure for me to introduce Professor Luiz Eugênio Melo. This is the new scientific director of FAPESP, and will give some words of welcome. Professor Luiz Eugênio.
Thank you, Julie. Good morning, you all. It's a pleasure to be here on this yet another webinar from the BIOTA program. The BIOTA program is one of the most successful programs FAPESP currently has. It's on its 20th anniversary. And today's topic is of critical importance. It's, of course, quite relevant for a country in Americas where megafauna, as we understand, has been partly extinguished by original settlers that likely came from Asia. It's, of course, of foremost importance for us to understand how the different species evolved one from another. And part of that relies on being able to adequately understand to which orders or families each species derives and it's linked. In addition to that, of course, it's of importance to understand the spreading nature for different seeds and plant species to which those animals contribute. So that's just to highlight the importance that we at FAPESP see from not only the BIOTA program, but also from today's webinar. I would like to once again express our gratitude to Professors André Freitas, Tim Newbold, and Thomas Lewinsohn, today's speakers, and give back the word to Julie. Thank you.
Thank you, Professor Luiz Melo. And it's a pleasure for me to introduce Professor André Freitas. André started in the BIOTA program as a postdoc student, and now he's leading large projects. And currently, he is also the director of the Biology Institute in my university. So he's my boss. So, André, the floor is with you.
Thank you very much, Carlos Jolie. So good morning to you all. Good morning, Luiz Melo. Good morning, Thomas and Tim. Good morning, the staff of FAPESP that's making this possible. I will do a short presentation discussing a bit of the results of the taxonomy and systematics results in the context of the BIOTA. As Jolie mentioned, I am myself a son of BIOTA program since I started my career as a postdoc in a project completely linked and directly linked to this program. I'm sharing my screen. 1 minute. Okay. So, as a son of Biota linked to natural history and systematics and taxonomy, I'm very happy to have the opportunity to do this presentation. So, I think it could be considered that's not needed to say that taxonomy and systematics are absolutely important, not only for things like knowing our biodiversity, or also because they are basis for conservation projects, but also for several other areas of investigation, such as biological control and biological education and human health. But that was not the feeling at the end of the 90s. Actually mentioned, when I was still a PhD student in 1998, we are living some bad times for people like me that were interested in taxonomy and natural history. Most of my, not most, but some of my colleagues talked, "Ah, you like taxonomy. I'm not sure if you'll get a job, or if you like natural history. This is not as fancy as let's say high ecology." At that time, or some years before, some important and influent authors have written papers discussing the fate of the taxonomies and systematics. And also, I remember this very nice editorial of Douglas Futuma that came on 998, discussing about the life of the naturalist. Curiously, it was in the American Naturalist Journal that at that time it was become, in the vision of Doug Futuma, not very well a good natural history journal. And so the BIOTA-FAPESP program came to completely change this, starting in São Paulo and then basically spreading to all Brazil. The program started in 1998, and that was a new hope for all those people like me that were interested in biodiversity. And at first, 11 projects were submitted in 1998. Most of these approved in 1999 and 2000. And basically most of them, or virtually all of them, focused in some of these issues, such as taxonomy, systematics, and biodiversity knowledge gaps. So let's say they were focusing on doing inventories in some critical and knowledge gaps in São Paulo state. And that program, that initial set of 11 projects, they were crucial for expanding the number of the taxonomies in the following 2 decades. That was really, really important, because together with the expansion of this taxonomic expertise, the BIOTA-FAPESP left a clear signature in science in the following years. And just giving some examples, one from my study group, the butterflies. This is a very important hyper-diverse butterfly group in the Neotropical region, the Euptychina. It's a group with more than 400 species and dozens still of undescribed taxa, including some complexes of cryptic species. And because they are not that bright colored, they were what they call these boring brown butterflies. They were ignored by taxonomists in the last century. And as a simplest cartoon to show that, after the studies of Butler and Felder and Weimer by the end of the 9th century and beginning of the 20th century, the bars that show the number of described taxa clearly show that by the end of the 90s, start of 2000, the number of taxa described by projects linked to Biota definitively puts Brazil and São Paulo in the scenario of taxonomy advances in this group. So as you can see, from 2000 to 2010 and 2011 to 2020, the contribution of Brazilian researchers to the knowledge in this group were enormous, and these were virtually absent before the Biota project was have started. Similar results we can find in endurance. By the beginning of the Biota program, 108 species were known in the state of São Paulo, and today this number have increased to 250 species, or let's say it's about 70 new species described. And more than that, circa of 150 new species are described, including other states and biomes. And this includes some very interesting groups, such as the Phrynomedusa. That's a group of species with high potential to have bioactive compounds. There are other groups not as fancy as butterfly or frogs. Let's say the Gastrotricha, a small phylum of male fauna organisms, very hard to study because they have many technical problems to be collected and fixed. It's a challenge to have types for these groups. And no new Brazilian species have been described since the 90s, when a Polish scientist comes to Brazil and described, let's say, 20 species. So recently, the Biota supported this. That's the only research group studying this phylum in Latin America by Professor Garrafoni at UNICAMP. And in 4 years of a project that just started, let's say, 9 new species are described and a dozen new species are waiting to be described in the next years. So what's the Biota legacy for taxonomy and systematics? I think this is important to highlight. The first is the infrastructure. Biota gives a possibility of creation of new collections in some institutions in São Paulo state, but also helped with improvement of those previously established collections. Also, Biota increased the number of equipments available for researchers and the establishment of facilities for taxonomy and systematics in São Paulo. Also, data viability was increased because museum collections could be improved with all these new field trips financed by the Biota projects, and that included now lots of material for some poorly known regions in São Paulo, in some neighbor states, and even another neighbor countries in South America. Also, we have consolidation and publication of large databases, and for sure, the electronic and virtual materials also available. And more important than that, the formation of human resources. So since Biota program was settled, we just helped for the formation of a whole generation of systematists, which ultimately resulted in a plethora of taxonomic publications from several taxonomic groups. To have an idea, a simple search in Web of Science, including terms such as new species, biota, and zoology, gives us an idea of the number of published papers on several taxonomic groups that mostly reflects the projects financed by the Biota. Of course, there are some well-known groups for being well represented by active research groups in São Paulo, such as Lepidoptera and Diptera and Crustacea. But it's good to highlight some groups that previously were not highly studied, such as Acari from bird feathers, and some groups of male fauna animals, such as the Gastrotricha and Kinorhinca, and other interesting groups for which we have very low information on number of species and identities of species in São Paulo and Brazil. So there's a large room to Biota to expand this. So we can maybe in the next 10 or 20 years have a good feedback of new species from groups such as bacteria, fungi, Quanoflagellata, and another male faunal groups, or ignored and neglected groups of arthropods. So almost at the end, I would like to call attention for a noteworthy detail. That's the persistence of the taxonomy knowledge. Basically, I believe that resources that are applied in natural history and systematics are always well-used resources. Why? Because a characteristic of this knowledge, that's its persistence. Collections and specimens, for example, it's kind of a science that's eternal. They will be useful and studied forever if they are well preserved, of course. The half-life of taxonomic and natural history studies is usually longer compared to other areas, such as the molecular areas. So species descriptions, taxonomic revisions, and natural history data, all these are relevant for longer periods, but really long periods. It's easy to get an example. The Linnean types are still relevant for us. And good taxonomic revisions, a generic review or something like that, they are used for decades. And I'm not saying that the molecular and technological science is not relevant. Yes, a lot of. But it's important to say that for every real or dollar that we apply in taxonomy, this will be still useful after a century. The same for equipment. To have an idea, a good 40 years old or 50 years old stereo microscope is still useful today. Of course, it's not as good as the last releases, but they are still useful and provides excellent data for taxonomic studies. So in short, the financial research applied in taxonomy will revert in good science for longer periods compared to the most technological areas. And we need to use it. So our resources now more and more scarce for taxonomic and systematics, for sure. And we have lots of studies showing that well-maintained natural history collections and taxonomists will help to explore and do prospection of our diversity, especially in places like Brazil. The future of tropical ecosystems depends on this good taxonomy. And recently, very recently, in fact, by the end of September, a very interesting paper discussing some of the topics I discussed on my presentation now comes to light. It's a very interesting. I recommend you to read it. And we have no doubts that we need to use our resource for taxonomy and systematics, as well as to other important areas as health and technology. So what could we think about the next 20 years? Thank you very much to the coordinators of the projects that send me some ideas and insights about that. I tried to do a very short consolidation of this, and I found 8 key points that should be highlighted from these leaders of the Biota projects. We need to sequence complete genomes for as many species as possible. This includes type specimens, what we call now museomics. It's a very interesting and expanding area. Institutions should promote the hiring of taxonomists, of course. We need to increase more and better scientific outreach. This is really important now. We need to show to the society what we are doing. We still need to stimulate biological inventories, even in, let's say, well-known, but not that well-known states as São Paulo. Increase interdisciplinary initiatives that include taxonomy and systematics. We should promote what we are calling integrative taxonomy. This is to deal with different sources of data. The implementation of electronic collections made available in websites, all types and all relevant information, and provide a broad menu of workshops, training courses, and another opportunities for students in your sites interested in taxonomy and systematics. So I'd like to thank you, these three girls that helped me with presentation, organization of data and graphics, to Carlos Joly, Thomas Lewinsohn, and all project coordinators that contributing by sharing data, ideas, and information. Thank you for all these agencies that helped our research in the last 20 years. And now I finish my presentation and we'll pass now it's with you, Thomas. And I hope we can have more questions by the end of this. Thank you very much.
Well, can you hear me?
Yes.
Okay, so good morning to everybody. Thank you, André, and thank you especially Carlos and the team at BIOTA-FAPESP for this invitation, having participated in the program since the preliminary discussions when we were conceiving this program. It's a special honor to be invited to participate in the celebration of 20 years anniversary, and also in the taking stock of what we have accomplished. So I hope the screen is on. Can someone confirm that the screen is visible?
That's okay.
Okay, so right. So the first question is, what's the scope of this presentation? So initially I was invited to an overview of animal ecology, but so many projects have intersections with other areas, with other groups, that I decided to put animal ecology, strict animal ecology projects in the perspective and the broader perspective of ecology and biodiversity projects in FAPESP. So in the strict level of what would be labeled ecology projects, we would have 40 projects in 20 years. But if we include other projects which have ecology and biodiversity in their titles or keywords, we come to 107 projects. And this is the set of projects that I will be considering to discuss, to do a brief overview in which I will profile this set of projects, which are basically regular projects, thematic integrative projects, and the Young Researchers Grants, which also have more or less the same standing as the so-called regular grants. So to profile them, I used three simple criteria, straightforward. What is the main taxon of a project, or what is a combination of taxa that the project focuses on? Then the main habitats, the main habitat included, and then what is the key goal, the chief goal of a project, whether it's a serving or inventorying a group or a site, whether it's focusing on processes such as drivers for biodiversity, whether its chief goal is examining questions related to conservation or resource use, and finally, whether it's data integration and GIS and Atlas projects. So in considering this set of 107 projects over 20 years, I also decided to give a larger weight to thematic projects because they last longer and they include more people. So they have larger research teams. So giving them a weight of 3 compared to regular projects and young researcher projects, what we have is a clear prevalence of projects focusing on plants, and then not surprisingly terrestrial vertebrates and terrestrial arthropods. What I'd like to highlight here is that we have a concentration on, and these groups, they're largely terrestrial. We have a fairly even division between freshwater and marine projects in other groups. And we have relatively few projects on microbes, and including dual taxa, which are largely terrestrial. We'll get back to this in the next slides. If we look at the main goals of the projects, we have over these 20 years a concentration on serving, inventorying, and describing species. So this ties in with what André showed us. So this is an obvious prime task for programs such as BIOTA. And what is interesting is that relatively fewer projects focused on intensive work in one locality, and most of them had a broader regional landscape to continental scale. Also, on different use projects which focus largely on conservation and use of natural resources, these are, there's a slight concentration in projects in terrestrial and marine, and a lack of these in freshwater habitats. I would like to call attention that there are relatively few, relatively slight effort on restoration and ecosystem services and socio-ecological aspects still. And this obviously calls our attention as one of the areas which would be highlighted in the next 20 years. The biodiversity drivers is a surprisingly low number of projects try to identify drivers for biodiversity. More of an effort was spent on ecosystem processes, which we saw in a previous webinar, and relatively small number of projects focused on interactions, but these are largely terrestrial. If we look at this in a different way, I would like, if we look at different taxa and the goals of the projects associated with them, I would like to call attention to the fact that one thing which surprised me is that projects focusing on microbes were largely descriptive of microbiota and relatively few, a small effort on processes, which, given the importance of microbes in a number of functional areas and ecosystem processes, is something which again begs for further work in forthcoming projects. And again, we have a prevalence of projects on plants, but these are fairly evenly distributed among the different major goals that I identified. So this gives us an idea of where we stand with projects on animal ecology, what the spread is. It's not quite even, but there is a fairly even distribution of effort, but obviously there are some groups which would benefit from more work in the near future. Finally, here in this profiling of projects, I would briefly show just the combinations of taxa which were studied in projects on interactions. Again, I have to remind you that this is not 9 projects, but these give a greater weight to thematic projects. So we have a concentration of projects of which investigated arthropods and their relations with microbes and plants and insects largely, and a fewer projects on other areas, as a minor focus here on algae and associated marine and freshwater invertebrates. So the next step is to take a quick look at results. And I will just bring up, this is a sample of studies which have been presented. And of course, whenever you make a choice to highlight one or another result, you make more enemies than you lose friends, so to speak, because of course people would say, why was this paper highlighted? We could spend at least an hour picking up remarkable results which have been published over these 20 years. So this is just a sample which represents the quality and diversity of results that have been produced over these 20 years. So when I'm talking about surveys, I don't mean only species surveys, species lists, far more than that. So this is well exemplified by this study by Celiodard and co-authors on reproductive modes of frogs, which is one of the bestsellers of the publications resulting from the BIOTA-FAPESP program. It's highly cited. And another typical example here is a study of termite diversity over a gradient, a latitudinal gradient in Atlantic forest. So of course the limits, as André already mentioned, and we saw in previous webinars, BIOTA-FAPESP is not restricted to the state of São Paulo by any means. So other kinds of assessments also already are directed at applications. So this study on freshwater fish is the production includes the production of an index to measure integrity and responses to various kinds of environmental disturbances. So this is highly important in terms of assessing of biodiversity assessments and environmental assessments. Likewise, this study is in urban spaces in the city of São Paulo. It focused on mosquitoes, and it used an interesting approach. Rather than just making lists of species, it did an island biogeographic approach. So here, each line, each row is a park in an urban park in São Paulo, and these are species, mosquito species found here. And an interesting commentary on this study is that the metric used to analyze these data was itself derived from by Almeida Neto and Guimarães and co-authors within another BIOTA-FAPESP funded study. So here the method is also a result, the method itself employed in this study is also a result of further study within the BIOTA-FAPESP program. Leaving aside all modesty, I'll briefly mention here a paper that was a direct result. This is a theoretical study on interactions, on patterns of interactions. And again, this is one of the, has been quite successful. It's nearing 300 citations now, and it's being applied in a number of areas which go way beyond plant-animal interactions only. Another example of a theoretical advance is this study by Renata Pardini, Jean Pomecka, and other co-authors, which developed a new model, a threshold model, to investigate diversity and processes in remnants, in fragments, especially forest fragments, but it could also be applied to non-forest fragments of various sorts. This model is quite successful, and it's being very fruitful, and certainly will be applied for a long time after in the near future. Another focus, I would say, of results is are the studies on interactions, and they are exemplified in by two studies here. Marcio Lambert's led study on bacterial diversity, which opened up an entire new front of investigating diversity within the Atlantic forest, and at the other extreme, as an outcome of extensive work especially in Cerrado, in the Cerrado biome, Paulo Oliveira co-edited a worldwide overview of ant-plant interactions and their impacts on various terrestrial ecosystems. So this brings us to a further scale of results which go way beyond the scope of the BIOTA project itself. Interaction patterns were also examined in dynamical models, such as this fairly recent publication on fruit frugivore interactions, which are being modeled as a meta network, which is a very recent approach. So we're incorporating state-of-the-art modeling and developing original modeling at the same time as we produce the data sets to which these can be applied. And these lead straight into applications, which are exemplified by one of the results. There's another focus on pollination is a hot theme within the BIOTA-FAPESP program. And the results are not only serving pollinator-plant interactions, but in this case, analyzing, predicting shifts in pollinators with climate change. So this is a highly important key application of results. That's just one example of several projects which have been successful in developing these new lines. We'll return to this in the final slides. And as a final view on the results of the BIOTA projects, scientists from the program have been contributing to worldwide assessments. I have highlighted here in green within this huge set of this worldwide estimate of the number of tropical tree species. There are many participants from senior scientists to postdocs, which contributed to this, and they contributed in two ways. One, with data, so they contribute to a global data set, but also with ideas and knowledge, so that this insertion in international science is certainly one of the major results of the BIOTA-FAPESP program. And of course, we should here mention the participation of a number of investigators from the BIOTA-FAPESP program in the international panel on biodiversity and ecosystem services with major contributions. And I had to bring this up and remind us that, in fact, the IPBES platform has been proposed as a proposed candidate for this year's Peace Nobel Prize, and we're all, of course, rooting for them. So let's go to a balance and prospects. What are the key achievements of the BIOTA program in this set in this overview that I presented very briefly to you? First of all, I'd say that we scaled up research in ecology and biodiversity. Many experienced scientists were engaged in this in the program from the start. Some of them had decades of work, but they worked largely individually or in small groups with the students. So there was a learning of organizing into bigger, not big, but bigger science, working together, which enabled a number of them for the first time in their professional careers to tackle larger questions, which this required learning how to organize teams, organizing equipment and logistics, integrating people, integrating their egos. All of this doesn't come spontaneously. There's a lot of learning involved, and I think that the BIOTA-FAPESP program was absolutely essential in bringing scientists to work together in a different way. The other point that I'd like to make is that we had quantum advances in important areas. Obviously, we had huge advances, especially on the Atlantic forest and Cerrado, which are the major biomes represented in the state of São Paulo, and coastal ecosystems as well. But as we've seen in previous webinars, results extend from the Amazon to the natural fields in Pampa and Rio Grande do Sul. So we had quantum advances in understanding of diversity and community structure in a number of groups. And we started investigating key drivers and processes in ways which had not been attempted before. And as I highlighted, this goes parallel to developing new theory and methods. And all of this leads to the fact that Brazilian science and scientists engaged in the BIOTA-FAPESP program have achieved a level of international recognition and insertion, which is way beyond what we had 20, 30 years ago. But looking forward, where do we stand now? First, unfortunately, it's quite clear that the situation that we have in 2020 is way worse than when the program started 20 years ago. We have a multi-scale crisis, of course, in the environmental crisis, plural, are much worse now than they were 20 years ago. We're closer to yet unknown tipping points. But at the same time, the financial constraints now are worse everywhere, but particularly in Brazil, than they were 20 years ago. And we have a serious political crisis at all levels, from the national government to the state government. There's a clear hostility to science and government, which means that not only do they want to direct scientific funds to other goals, but they want to move them from science to other politically motivated applications of financial resources. So there is a clear move both on the national and the state level to cut back on financing for science. Countering this, what can we do to counter this? First, defend freedom of investigation, which is under serious risk. This is a very important matter. I won't go into details, but I think that's a starting point. The other thing is we have to work better together with groups in other Brazilian states and overseas as well. We do have to enrich our interchanges. It's not communication, but interchanges with citizens, with journalists, and with serious politicians, to give them a clear idea of what we are producing and what the use of this knowledge are. And we have to be especially alert to novel projects which will address new social and economic demands. And this brings me to my final slides. I would suggest that we focus on key issues. We reflect on key issues. One of the ways to do this is do a horizon scanning exercise on identifying new and impending issues, and then designing research, creative research to address these. Focus on certain drivers which we know are hugely important. I mentioned this briefly before. So plant microbe interactions are hugely important for ecosystem processes. I would suggest that we work with leaders in agricultural business to focusing research on new production systems, such as precision agriculture, to see how they impact the environment, whether they in fact are more sustainable. We need critical and independent assessment of sustainability in many ways. And as a final suggestion here, which comes from the researchers which responded to our questionnaire, as well as ourselves, so incorporating new tools such as genomics and isotopes in surveys or to trace ecosystem pathways will allow us to tackle new questions or enlarge the scale of our work in ways not considered possible 20 years ago. We need we need to fill some gaps, critical gaps in understanding geographical distribution of organisms. And we need more work on historical ecology, in which we compare previous states with current states. Sites which have been well surveyed before, such as the Boracéia station, should be resampled with comparative methods so that we can see what has been lost and gained in terms of assemblies and species lists. And multi-taxon assessments are obvious, but I would like to emphasize in closing that we do need long-term observatories of biodiversity and ecosystem services so that we can track changes and make assessments on how fast things are changing and whether we're able to reverse negative trends in any way by new environmental priorities and policies. So I thank Carlos for inviting me, to give me the opportunity to participate in this celebration. The team, I thank especially the colleagues who did respond to our questionnaire. And I thank everybody who in the current Brazilian situation stands up for free thought and sound science. Thank you very much. And back to you then, Carlos.
Well, thank you very much, Thomas and André, for the very nice presentations you made. I forgot in the beginning to introduce Professor Thomas. Professor Thomas is a retired ecology professor at the animal biology department from the University of Campinas, and has been working with this pushing forward the ideas in terms of theoretical and using different groups of animals to advance on the theory of biodiversity, different theories, and has a lot of cooperation with international groups. So I thank you both of what the BIOTA program has produced so far. And now it's a pleasure for me to introduce Professor Tim Newbold. Tim is based in the Center for Biodiversity and Environment Research in the Genetics, Evolution and Environment Department, University College London. And his group is researching with the aim to understand how biodiversity is changing in response to recent environmental change. So, Tim, it's a great pleasure for us having you here in this BIOTA webinar. The floor is with you.
Thank you very much, and thank you for inviting me to come and give a talk to you today. It would have been nice to meet you all in person, but in these strange times, at least I can give this talk online. So today I will present some recent work that we've been doing trying to understand the impacts of land use change and climate change on biodiversity. And today, particularly, I will focus on some very recent work that we've been doing on insects. So before I start, I'd just like to say a big thank you to my wonderful research group at University College London, who have provided such important inputs to everything that I will present today. And also, I'd like to acknowledge funding, particularly from the Royal Society, also from the Global Challenges Research Fund and the Natural Environment Research Council here in the UK, also the Lewinsohn Trust and the Economics and Social Research Council. And I'd also like to thank some tremendous collaborators who, again, without whom the work that I present today wouldn't have been possible. And particularly today, I'd like to express my thanks to Georgina Mace, who was a giant of conservation biology and sadly, tragically passed away a couple of weeks ago. So in recent months, we've seen some big reports coming out on the status of the world's biodiversity. So just a few weeks ago, the latest Living Planet report from WWF estimated that populations of the world's vertebrate species have declined by around 68% since 1970, so over just under 50 years. And the first global assessment of the Intergovernmental Panel on Biodiversity and Ecosystem Services that was published last year estimated that as many as 1 million species worldwide may be at risk of imminent extinction, and around half of these were invertebrates. And of course, these losses of biodiversity are being driven by massive increases in the impacts of humans on the environment, and in particular, vast increases in the amount of land that we're using, particularly to grow our food for agriculture, and also for other human uses, such as places to live. And also, of course, increasingly by emissions of greenhouse gases and the consequent changes in our global climate that we are seeing. But we know that not all species are impacted equally by these changes to the environment. So, for example, here on the left, there's a picture of the Bramble Cay mosaic-tailed rat, which was found on a tiny atoll off the coast of Australia, and is thought to be the first mammal species to have become extinct as a result of climate change. And on the right is the comma butterfly, which at least here in the UK is spreading rapidly and seems to be benefiting mostly from changes to the climate here, to warmer conditions. And at the same time, what we know about biodiversity, the data that we have on biodiversity is very unequally distributed. So shown here, the green bars, the heights of the green bars represent the numbers of species thought to be found within different groups. And the orange bars, the height of the orange bars represents the number of data in the Global Biodiversity Information Facility, which is one of the biggest databases on biodiversity in the world. And what you can see here is that the vertebrates on the far left, whilst they account for a very small fraction of the world's biodiversity, have by far the largest share of the data that we have. And of course, we saw that in Thomas's talk as well earlier. And it's great to see that the BIOTA program has been doing so much to increase the representation of the less well sampled biodiversity. And so the invertebrates here in the middle, whilst accounting for a large share of the world's species, have a much smaller fraction of the data. And we know that this poorly sampled biodiversity is also vitally important. So just to take the invertebrate macrofauna as an example, so here the arthropods, we know that they contribute a number of vital functions. They perform roles such as pollination, predation, and cycling of nutrients. And I should acknowledge at this point that, of course, plants also provide vital roles within ecosystems, but as a zoologist, my focus tends to be on animals. And we're beginning in recent years to get more data on invertebrates and insects, including insects. And it's becoming clear that we're seeing large declines, particularly large declines, it would seem, in invertebrate, in insect populations. And so, some of the science that we've seen come out in recent years has been challenged by the patchiness of the data that we have, and some of the media coverage has perhaps been a little hyperbolic. But certainly, if we look at the evidence that's now amassing, so this lovely synthetic paper by Van Klink et al. That came out earlier this year amassed all of the data that could be found on trends in insect abundance and insect biomass over time. And what they found was that, at least if you look at terrestrial insects, that there are more cases of insects showing declines than insects showing increases. And on average, they estimated that across these different studies that have looked at insect trends, that on average there were declines of around 10% per decade in insect abundance. So when we know what it is that's threatening biodiversity, so particularly when we look at the vertebrates that are very well studied, and we look at the primary threats to those species, what we find is that in most cases, land use is the major pressure on changes in land use are the major pressure on species. At the moment, climate change corresponds to is responsible for a relatively small fraction of known primary threats to vertebrate species, but of course that's expected to grow as climate change accelerates in the coming years. If we look at how much these different pressures on biodiversity have been studied, what we see is the reverse. So the effects of climate change have been studied far more often than the effects of land use, at least here when looking at large-scale continental or global studies, not at the sorts of small-scale studies, as I'll come on to in a minute. So in the first part of this talk, I will present some of the work that I've been doing over the past 8 or so years, trying to fill that gap in global scale studies of changes in biodiversity. So looking at how globally land use has impacted biodiversity. And this work started really with the Predix project that is led by Andy Purvis at the Natural History Museum in London. And this project was motivated by the observation that the scientific literature is full of studies that look at the local scale impacts of different types of land use on biodiversity. And so the idea with the Predix project was to gather together as much of this data as we could, and to then build up a picture of how, on average, land use impacts biodiversity, different types of land use impact biodiversity around the world. And this project was made possible thanks in large part to hundreds of field researchers who were willing to share their data with the project. And of course, that itself was the work of those individual field researchers was underpinned by the sorts of taxonomic and natural history skills that we heard about in the first talk in this webinar, and the sorts of skills that the BIOTA program has done such a great job of promoting. By the time we published the Predix database, we'd been able to amass just over 3 million records of biodiversity around the world in different types of land use, which is far fewer, of course, than things like the Global Biodiversity Information Facility, but we had a much more restricted focus here. And particularly, we needed studies that compared biodiversity in different types of land use in a controlled fashion. And these data were drawn from around 26,000 locations around the planet, from 94 of the world's countries, and representing just over 47,000 of the world's species. That's around 2% of all species that have been described by science. And as you might have seen from the map, Brazil was particularly well sampled, and I think that's thanks to programs like BIOTA that have done such a good job of collecting these sorts of data. We published the Predix database in the first release of the Predix database in 2017. It was published under Creative Commons license, which means that it's free for anybody to use, including for commercial uses. So you can go to the portal of the Natural History Museum and download the data. And the gargantuan effort of collating and curating all of those data it is a testament really to the work of my colleague Lawrence Hudson, who designed the database and the mechanisms for bringing together the data, checking it, and then ultimately sharing it. A big focus of the Predix project, as well as trying to get data spreading across as much of the world as possible, we were also aiming to get data spread across the major groups of animals and plants, and to be as representative of those groups as we could. So shown on this graph here, along the bottom, is the number of species that have been described, so the number of species known to science, and along the side, the number of species that we captured within the Predix database. So anything that falls above the diagonal line, any groups that fall above the diagonal line are groups for which we were able to gather data for more than 1% of all species known to science. And we were fortunate to be able to achieve this level of coverage, not only for the vertebrates, which was relatively easy, but also for the world's mega diverse plant and invertebrate groups, particularly the insects and the arachnids. And so to be able to build up a general picture of how different types of land use impact biodiversity around the world, we had to divide those types of land use up into broad categories. And so we recognize 6 broad categories of habitat. So pristine or primary habitat, habitat recovering to a natural state after past destruction, or secondary habitat, and then 4 classes that are currently used by humans: plantation forests for woody crops, herbaceous croplands, pastures used for livestock grazing, and also urban settlements. And within each of these 6 broad categories, we also distinguished 3 levels of the intensity of use by humans. So, for example, in natural habitats in primary and secondary vegetation, that could describe activities, increases in the intensity of activities such as bushmeat hunting or selective logging, and then in, for example, croplands, describing inputs of chemicals, pesticides and fertilizers, and also the diversity of crops. So with more intensive land having monocultures, so lower diversity of crop types. What we found with the early work of the Predix project, where we looked at general patterns in the way that different types of land use impact biodiversity, was perhaps not surprisingly that where humans use the land, we have fewer species, that when we use the land more intensively, where humans use the land more intensively, we see further reductions in species, and that we also see reductions in species where there are higher densities of human populations and where locations are closer either to roads or to human population centres. And in the most disturbed habitats, so where there's the highest density of humans, closest to roads and population centres, and where the land is used most intensively, we saw declines of up to around 3/4 on average in the numbers of species. Now, of course, ecosystems are in reality very complex webs of interacting organisms. And so just looking at overall changes in the number of species doesn't give us a very good idea of the health of those disturbed ecosystems. One of the ways that we have begun to think about how environmental changes, how land use changes might be impacting the health of ecosystems is to classify the species within those systems into broad types, broad functional types, so separating species according to how they fit within the structure of the ecosystem. And so in a study that we published earlier this year, we divided species into these broad types. So we divided species according to what they eat, so whether they are predators, herbivores, or eat a mix of plants and animals, by their size, because we know that body size is very important for determining your role within an ecosystem, and also by whether species are cold or warm-blooded, whether they're endotherms or ectotherms. And then we looked at how sensitive these different groups are to land use changes, to the sorts of disturbed land uses, so agriculture and human settlements. And this was work that was started by Laura Bentley at the University of Cambridge as part of a student research project. And what we found was that generally predators were more sensitive to disturbed land use, so to agriculture and settlements, than species that eat plants or species that eat a mixed diet. And particularly we found that small predators are particularly sensitive. So, for example, things like arthropod, insect predators. So in recent years, two main areas of focus have been partly and overlapping areas of focus have been partly to think about how climate change might add to the impacts of land use change on biodiversity, but also to focus in a little bit on those species that appear to be particularly sensitive and also that are that fulfill particularly important roles within ecosystems. So in the past few years, we've been focusing particularly on insects. So some of this work has been a project on which I've been collaborating with colleagues at the University of Ottawa in Canada, looking at changes in the diversity of bumblebees across North America and Europe. And so in some work that was published earlier this year, work that was led by Peter Charoy, a PhD student at the University of Ottawa, looking at changes in the occupancy and in the range sizes of bumblebee species across North America and Europe. And what we found in that work was very large declines in bumblebees across both North America and Europe, but particularly in North America. And also that those declines were driven primarily by climate change. So it's interesting that bumblebees appear to be more sensitive to climate change than they are to land use changes, at least the sort of broad conversion of habitats from natural habitats to farmland. And this was the first time that there was a really clear, it was the clearest signal so far that we've seen of climate change on recent declines in bumblebees. More recently, we've been trying to extend the insights that we've got from this bumblebees work, both across the whole world, but also across across many more insect species than just bumblebees. So from here on, I'm talking about work that hasn't yet been published. So I would appreciate, please, if you don't share this outside of today's webinar. This is work that was started by a student working with us at UCL, Peter McCann, and now is being led by Charlie Althwaite, who's a colleague of mine at UCL. And so for this work, we've been using the Predix Database again, which has samples on insects from across the world, and looking firstly at how land use impacts insect biodiversity. So repeating the sorts of results that I was showing you earlier, but focusing here on insects. And what we find here, interestingly, is that we see large declines in numbers of species. But what's particularly interesting is that we also see very large declines in abundance, which is not something that we found when looking at all species together using the Predix database. And this seems to correspond well with what other people have found. So these other insect studies that have suggested that we're seeing very large declines in the abundance of insects, which of course is very important because we know that insects provide that the important roles that insects play within ecosystems depends on them being very abundant in many cases. But the other thing that we're interested in with this study is how climate change is adding to the effects of land use changes. And so we've been working on developing a indicator to measure how much the climate has changed in the recent past in a way that is, important for biodiversity. And so specifically what we're interested in here is how much the climate has changed compared to how much the climate varies naturally year to year. And so we've developed an indicator of climate change, which builds upon what previous people have done, that measures the amount to which, the degree to which average temperatures have increased relative to how much the climate naturally varies year to year. And so what we find if we map out this indicator, so areas of orange on this map show places where average temperatures have increased such that the average year now has temperatures at least as high as what you would expect only in 1 in every 6 years under baseline conditions. So in other words, places that are pushing beyond, are getting hotter than we would expect naturally. And so while the absolute numbers of degrees of warming of temperature have been higher at high latitudes, it's in the tropics that we're seeing temperatures increasing most beyond what we would expect naturally, because naturally in the tropics, variation year to year is much lower. And what's really interesting then is when we look at how this measure of climate change and land use change combine together to impact insect biodiversity. And specifically what we find is that the biggest declines in insect abundance and also numbers of insect species, but I don't show that here today. The declines are largest when intensive agriculture combines with high levels of recent climate change. So in places that have gone beyond those, gone beyond natural variability, so there's areas of orange on the map previously, where that combines with intensive agriculture, we're seeing reductions of more than a half on average in the abundance of insect species within ecosystems, which of course has very important consequences for the ability of insects to provide all of those important functions that we know that they contribute to ecosystems and to the growing of crops in those very agricultural systems. And of course, these sorts of results become especially worrying when we think about what's going to happen in the future. So as you see on the map on the right here, which shows what the situation might be in the 2070s, approximately, under a business as usual scenario. So if we carry on as we are, we can expect many more places in the world, and especially in the tropics to begin with, that are going to be going beyond the temperature conditions that we see naturally. And of course, this is likely to have very important consequences for insect biodiversity and very important consequences for agriculture that's taking place there in future. So just to conclude then, it's becoming clearer as we get more and more data on insects that we're seeing very large declines in the diversity of insects within ecosystems and in the abundance of insects within ecosystems. And from some of the work that we're doing at the moment, it's clear that that's driven by a combination of changes in the way that we use the land and also climatic changes. And so as we see more land use change in future to feed the growing human population and accelerating levels of climate change, then that presents a rather worrying picture for the future of insect biodiversity and other invertebrate biodiversity and the impact that they play within ecosystems. Thank you very much for listening, and I'll pass the floor back to Professor Joly. Thank you.
Well, thank you, Tim. For a very inspiring talk and provocative. Now we move on to questions, and I would first ask André and Thomas if they have questions to make.
Let me just, okay. Tim, this was an extraordinary talk, and we would need several hours to work this over. Hopefully there will be further opportunities. I think that the first question that I would have is that from the analysis that you've developed, and given the indications that you have, whether you not you individually, but as your group, the entire group involved in the Predix effort, have considered setting up a network of worldwide observatories focusing on using a minimum common protocol and a favored set of taxa, let's say.
Yeah, that's a really good question. Thank you. So the short answer is no. So the Predix project was it's a relatively small project. It's like it's a standard grant here within the UK system, and so really the focus here was to bring together all of the data that we could find that exists out in the literature, and to make the best use of that we could. Now, that said, of course, what we'd really like is this sort of network of samples of biodiversity made in a common way, and sampling the taxa that we know to be most diverse, most important, and of course, something that so one thing I didn't mention in my talk was that what I presented and what the first phase of the Predix project focused on was where we could compare different types of land use in different locations. So using spatial patterns to try and predict what might happen through time. And of course, one of the problems with that is that we know that how biodiversity changes through time is more complicated, and there are lags and all sorts of things. And so something that you mentioned at the end of your talk was how we need more long-term monitoring, and that is definitely the case.
André, do you have a question?
No, thank you very much. Oh. Very nice talk, team. Congratulations.
Thank you.
I have a question here from Professor Cristina Miyake from University of São Paulo. Cristina led a very large project that was a joint call between the BIOTA program and NSF. And she says, if I understood well, unpublished results show that richness and abundance of insects was higher in urban areas than in highly impacted agricultural areas globally. If this is correct, could it be an effect of pesticides?
Yes, that's it's a very interesting question. I think it could be to do with pesticides. That's not something that we've been able to test yet. And unfortunately, in most parts of the world, we don't have good data on rates of pesticide applications in most locations. So it's not something that, unfortunately, that we're able to test directly, but that is certainly one possible explanation for the results.
One comment, Justin. Could it be also related to light pollution?
Yes, yeah, that's so you mean that the greater light in urban environments, perhaps you sample more insects because they would come to that light. I think that's certainly possible. I mean, of course, we're always challenged with these sorts of correlational studies in how much we can say about what it is that's driving the patterns that we see. I mean, there are a number of differences between urban environments and agricultural areas that could drive differences.
Team, I have a question myself. You and a group of researchers from which Georgina Mace was part of. I had very good relations with her as well. We worked together many times. You published a paper in the Policy Forum of Science proposing a biodiversity target based on species extinction. This raises some polemic, so I would like for you to talk a little bit about the paper and the idea.
Yes, yeah. We of course knew it would be controversial, to put it mildly, probably. So the motivation really behind this work was to find. It was not to propose a single biodiversity target, but to find a biodiversity target around which the public might rally in the same way as people have rallied around the 1.5 degree target for climate change. And from various discussions that we had, what we decided as a group was that the thing that resonates most with the public is the extinction of species. It's a much easier concept to communicate to the public, and I guess you see that in, for example, the coverage of the global report of IPBES that came out last year. That it presented lots of different statistics on the state of nature, and the one that's been by far the most covered has been this idea that 1 million species might be at risk of extinction. And so the idea behind this policy forum was to propose this sort of target that might rally people, that wouldn't certainly be the only one that we use in science, but that is easy to communicate.
Yeah, I personally I agree with the idea. We need things that you can translate to the common citizen importance and bring them to be behind the proposition and use as an index that it's fairly easy to understand. And but as I said, there's some controversy even here in Brazil. Coming back to your talk, Professor Roseli Tuan, she thanks and congrats you for the webinar, and her point of view, the research in taxonomy, ecology, biodiversity of organisms related to infectious diseases is underrepresented in most of those general ideas. And she asked this, the second part of the question is for Thomas and André, if they believe that this is a gap in the BIOTA program. So if you can answer first, and then I go to Thomas and André.
Yes, yeah. I mean, certainly, yes, I agree. There were still some massive gaps, and even within the Predix database, where we tried to be as representative as possible. Even if you look at the invertebrates, it tends to be the ones that are easier to sample, the macrofauna, things that live above ground, and so there are still massive gaps for below ground biodiversity, for the microbiota, and of course, they're tremendously important.
André and Thomas, you think this is underrepresented in the BIOTA program?
I would agree, yes. I don't think this is a historical contingency. This has to do with the interests. I think that realizing that diseases can usefully be investigated using ecological and evolutionary frameworks, putting them in an ecological and evolutionary framework is relatively recent. So yes, it's a gap, but I don't think that it's a conscious bias. It's more a question of a developing culture of the researchers themselves. So I would expect that this becomes a major focus in future work. So definitely it deserves to be a major focus.
Yeah, what I have to say is similar to Thomas, and also remember that diseases in some part are ecological in terms of how they spread and what is this, but only an interaction among some organisms. In terms of taxonomy, I talk about that in my class since I became a professor at UNICAMP, that we don't have enough people interested in natural history of any kind of microorganisms, and only focus on disease, pathogenic microorganisms. And we have this in a broad context. I think we should have more natural history linked to their effects. And also remember that some of these groups that are pathogenic have lots of species that are not, and these are usually neglected. I have now a postdoc studying the trypanosomatids from butterflies. It's amazing because are like hundreds of undescribed species, but usually the interest is about 2 or 3 that are important in terms of Chagas disease and another very few things. So when we could link all this in a broad context, I think this gap could be filled, but we need to have more people studying that. And I agree with Thomas. It's in part cultural, but there are other things to be put in context.
Can I add quickly to my
Yes.
previous response? I would just like to mention that the first generation, the first 2 generations of parasitologists in Brazil, they went on extensive trips into the interior. So some helminthologists, for instance, were accompanied Rondon in his first incursions into the Amazon, to Western Brazil. And they had a tradition of hunting large game, shooting down large animals, but then they had a protocol of dissecting them for ecto and endoparasites. So they did a thorough search of these large vertebrates, which they would hunt, and brought back an amazing quantity of material, which you still have in institutes such as the Oswaldo Cruz in Rio. So these were mostly doctors trained as doctors, but they followed this tradition as parasitologists to get as much information as possible. So you had everything from lice to helminths added to the collection. So this kind of tradition would be really interesting if people who are doing surveys with vertebrates would associate with and collect samples from tissue and blood samples, which nowadays could be routinely screened for signs or indicators of parasites, and they could be molecularly screened. So it's a question of integrating teams and making better use of whatever information you bring from the field. So it's largely a question of design and adding a little bit to the field effort with a huge return in terms of further information in the sense that the question was asked.
Okay. Well, I think we have to finish this webinar here. I would like to thank you, team, for a very inspiring talk. I've been following the Predix project since it started. I think is a model of collaboration that we can that we started already within the Biota and should be kept and stimulated in this research. And it was a very good opportunity to have you presenting this nice set of results. So thank you very much. If you want to give a final word, then I will close the meeting.
No, thank you for the invite. It was really great.
Well, we will finish this webinar, and I will remind the leaders of projects, Biota projects, that we have another meeting that will start at 11:15 to discuss within ourselves what would be the next steps of the Biota. So for the public in general, thank you very much, and pay attention on our schedule. We are going to have our next webinar that is focused on what the Biota produced in terms of education and communication on the 21st of October. Thank you all. Muito obrigado a todos e nos vemos no próximo webinar no dia 21 de outubro.